last class we have seen something about the cell structure and the basic unit of cell, the basic details of the cell we have seen. And today we are going to start the specific kinds of cells, and we are going to start with the prokaryotic cell structure. Okay. So what are prokaryotic cells? So as I told you in the last class, pro plus caryon. Okay. So pro refers to before or primary, and caryon refers to nucleus. So that means the prokaryote, the word refers to before nucleus. That means they have or they don't have the true nucleus. Okay. So in other words, we can say they don't have the nuclear membrane or the nuclear membranous structures present within the particular prokaryotic cells. Okay. So we are going to see the details of the structures. Before that, we will see something about the size, shape, and the different arrangement of the particular prokaryotic cells. So what is the range of the prokaryotic cells? We all know all the prokaryotic cells are in the microscopic structures. They are very minute structures. They will be around in the size range in between 1 to 5 micrometer in size. So what are the shapes? So as I told you in the last class, the prokaryotic cells are available in various shapes also. Okay. So we can see it in the microscope. We can observe the particular various shapes of the particular microbe. So rod shape. Okay. So the microbe, while you are observing under the microscope, you can see the rod. Okay. So rod like appearance. So we can commonly call these kind of bacteria or prokaryotes as bacilli. And some bacteria, they are in a spherical shape. Okay. So we are calling those bacteria as cocci. And some bacteria, they will be in a shape of the spiral shape, and we are calling those bacteria as spirillum. Okay. So there are three major shapes, and some bacteria they will be like comma shape. For example, we all know the cholera causing bacteria, Vibrio, will be in a comma shape. So there are so many variations in the shapes of the bacteria is available, the prokaryotes is available in the nature. Like that, there are so many arrangements also available in the nature. There are single living prokaryotes, paired prokaryotes, and clusters of prokaryotes, and groups of prokaryotes. There are so many arrangements also present in a nature for the particular category prokaryotes. So whatever may be the arrangement, you should remember one thing: they are all single celled organisms. They can live alone as a single cell. That point you have to remember. Okay. So what we have seen till now, there are various sizes of prokaryotic cells. There are various shapes of prokaryotic cells, and there are so many arrangements present in the nature for the particular prokaryotic cells. And now we are going to see something about the basic structural details with the help of this diagram, and then we are going to see the internal details of each and every part present within the particular prokaryotic cells. Okay. So here you can see the external of the particular prokaryotic cell contains three major things: cell membrane, cell wall, and glycocalyx. So with these three together, we are calling that as cell envelope. Okay. So what is cell envelope of a prokaryotic cell? That cell envelope which encloses or which covers the entire internal organization of the particular prokaryotic cells, and it is made up of three layers: cell membrane, cell wall, and glycocalyx. So within the cell membrane, there is a liquid matrix is called cytosol or cytoplasm. Okay. So within the cytoplasm, there is a major DNA content. Okay, genomic DNA content is in the form of nucleoid. So as I told you in the last class and in the beginning of this class, there are no true nuclear membrane for the particular prokaryotic cells. The nuclear material, the DNA, is in the form of the supercoiled structure. Okay, so actually what is the actual structure of the particular prokaryotic or bacterial DNA? It is a covalently closed circular DNA. Okay, covalently closed circular DNA. Okay, so it is a tube-like structure or your cycle tire-like structure. Okay, it is a covalently closed circular DNA and it is being supercoiled like this. Okay, so in a coiled structure like this, and it will be twisted around each other and it will form a supercoiled structure like this, and the final structure is called nucleoid. How it differs from the eukaryotic cells? There are no proteins involved over here as in the eukaryotic cells. We all know there are the major packing proteins called histones. In case of eukaryotic cells, that will pack the nuclear material or the DNA into the structure called nucleus. But there are no histones involved over here, but the DNA is being packed as a supercoiled structures called nucleoid. We are going to see the details later. Okay, so the point you have to remember over here, within a cytoplasm there are some structures of DNA material that's in the form of nucleoid. So other than the DNA, there are some other genetic material also present over here, nuclear material. There are RNAs. Okay, so RNA refers to ribonucleic acids. They are also present within the cytoplasm of the particular prokaryotic cells. And apart from the DNA and RNA, there are some other genetic materials also present in prokaryotic cells. They are called plasmids. So the plasmids are extra chromosomal DNA that may be present or absent from prokaryotic cells. Normally they are present. Okay, so we have seen one or two plasmids over here, but normally there will be at least hundreds of plasmids present in a particular single prokaryotic cells. So apart from plasmids, there are some inclusion bodies. They are the storage molecules present within a prokaryotic cells. Just remember, there are no membrane bound structures. There are no organelles. You have to remember this point. There are no organelles. So what are organelles? As I told you in the last class, organelles are membrane bound structures. There are no membrane bound structures or organelles present within a prokaryotic cells. Okay, so there are inclusion bodies. That means you have to remember, there are no membrane bound inclusion bodies. They are membraneless inclusion bodies. So what is the purpose of inclusion bodies? They are going to store some materials, and we are going to see the storage properties of the inclusion bodies later in this class. Okay, so apart from this inclusion bodies, plasmids, there are other macromolecular structures called ribosomes. So what are ribosomes? In some books, in some textbooks, they will be referred as the organelles, and you have to remember, ribosomes are not organelles. Okay, ribosomes they are not organelles, they are just a macromolecular structure. Okay, so just remember this point, they are not an organelle because they don't have any membrane surrounding the particular structures. So that we are not supposed to call these ribosomes as organelles, we have to call these structures as macromolecular structures. Okay, so we will see the details of ribosomes. What is the size and what is the Svedberg unit of the particular ribosomes in the prokaryotic cells. These details we are going to see in the later classes. Okay, so apart from ribosomes, we are going to see the cytoplasm is filled with a liquid, semi-liquid matrix that contains so many different types of proteins and a different number of proteins. Approximately 3000 to 5000 protein molecules. That means the types. And while you are dealing with the numbers, it will be around lakhs. Okay, so lakhs of proteins in the cytosol or the cytoplasm of the prokaryotic cells, and the number of the types of proteins refers to 3000 to 5000 different types of proteins in the particular single prokaryotic cells we can observe. Okay, so apart from this, within a cytoplasm we can see the structures, the invaginated structures of the plasma membrane or cell membrane. Those structures are called the mesosomes. Okay, so what are mesosomes? Mesosomes are the invaginated structures of the plasma membrane. So what is the need of mesosomes? Okay, suppose you have a cell in this size, and the next cell in this size, and the next cell in this size. So while we are comparing the size to volume ratio. Okay. So here this cell can absorb more amount of substances and exchange more amount of substances with the environment compared to this cell, because here even though the particular cell has a more amount of volume, the surface to the volume ratio, surface to volume ratio is very low compared to the particular smaller cell. So what it means, it should have the larger membrane or surface structures, then only it can absorb the more amount of substances from the environment. So to increases the surface to volume ratio, the prokaryotic membrane has some invaginated structures like the mesosomes. There are so many purposes for the mesosomes, and we are going to see those purposes later in this class. Okay, so apart from mesosomes, so as I told you, there are cell membranes. So cell membrane is made up of phospholipid bilayer. So basically they are lipid compounds. So just behind the cell membrane we can see the cell walls. So cell wall is made up of the murine compounds, and we are going to see the details of all the compounds present within the particular, all the biomolecules present inside and outside of the particular prokaryotic cells. And just behind the cell wall there is a structure called glycocalyx. Glycocalyx is made up of normally capsules or the slime materials. Okay, so apart from these three structures, there are some other structures also present outside the particular bacteria. They are called flagella, fimbriae and pili. Okay, so all the three things, fimbriae, pili and flagella, are made up of proteins. So we all know what is the role of flagella. The flagella is for the locomotion or the movement of bacteria, and fimbriae is for the attachment of bacteria to the surface. It may be to the host or it may be to any surface. Then only it can attach to the particular surface firmly. And pili is a sex apparatus for the bacteria. Only the conjugation, the conjugating bacteria will have the structure called the pili. Pili is actually a form of fimbriae. Okay, we can call that as sex pili. That means they are going to participate in the sexual exchange of the genetic material in between the bacteria by means of the process called the conjugation. Okay, so these are the three structures, the external appendages present in a particular prokaryotic cells. Okay, so this is what we are going to see today. So what are the things present in a particular prokaryotic cell? So what are the things we are going to see? We are going to start with the cell envelope. In the cell envelope we are going to see what is cell wall, what are cell membranes, and what are glycocalyx, and what are their functions we are going to see. Then we are going to see the external appendages, what are flagella, what are pili, and what are fimbriae, and what are the different types, and what is the particular role of the each and every structures present in the outer surface. Then we are going to enter into the cytoplasm. Then we are going to see the what is nucleate, how the structure is being arranged, and what is the role of the DNA, and what are the other genetic materials like RNA and plasmids present in a particular prokaryotic cells, and what are ribosomes, and what is the size and shape and functions of ribosomes for a particular prokaryotic cells. That is what we are going to see today in the class, today's class. Okay, and now we are going to start with the cell envelope. And as I told you, cell envelope contains cell membrane, cell wall, and glycocalyx. Okay, so we will start from the outermost layer called glycocalyx. So glycocalyx, as I told you, so among the three layers, it is the outermost layer present in the particular cell envelope. So normally glycocalyx is made up of carbohydrate materials. Okay, so there are so many variations also present. Normally they are made up of the carbohydrate materials, and normally they form a structures called capsules. Okay, so just remember that not all bacteria or all prokaryotes possess the particular glycocalyx structures or capsules. Only the few bacteria which can infect the animals, they will have the particular capsule like structures. Okay, so what are capsules? They are the covering present the outermost surface of the particular prokaryotic cells. So what may be the role of the particular capsules, they will give the protection from the phagocytosis, and they will give the purpose of attachment, and they will prevent the bacteria from drying. Okay, so these are the major roles participated by the particular capsule structures or the glycocalyx structures of the particular prokaryotic cells, and they are infective. As I told you, they will give the resistance to the bacteria, so that they are infective particles for the animal beings. Okay, so these are the purposes of the capsules. So apart from capsules, there are some other similar structures, but not in the particular thickness of the capsules are also present in the bacteria, and those structures are called the slime layers. So what are slime layers? Slime layers also the layer present the external surface of the particular bacteria, but they are made up of thin layers of the particular carbohydrate. So what is the role of slime layers? Slime layers will form the biofilm. So they will form the biofilm. So what is biofilm? We can see the tooth caries and a dental plaque, and the yellow deposit we can observe in the infected teeth, and those appearance, the yellow color appearance, that slimy appearance is being produced by the particular the bacteria present on the surface. That layer is called the slime layers. So what may be the purpose of the particular biofilm? They are majorly, they are form the aggregates of the particular bacteria, and they form the aggregates and attachment to the particular surface. Unless you are rubbing it out of the particular surface, you cannot remove the particular aggregates and attached bacteria from the particular surface, and that is the purpose of the particular glycocalyx. So what is glycocalyx? Glycocalyx is the outermost layer present in the particular cell envelope group, and the role is majorly it is attachment, and they are providing the protection from the drying and providing the protection from the phagocytosis, and so that it can act as a infective particle for a particular bacteria. So the middle layer, the cell wall. So what is cell wall? So as I told you in the last class, all the prokaryotic cells and some eukaryotic cells, they will have the cell wall like structures. And what is the role of cell wall and how it is made up of, and what is the material will make up the particular structures. So what is the role of cell wall? That will give the protection. So as we can observe, the prokaryotic cells normally live in an environment, they will have the different solute concentration. So what may be the solute concentration compared to the environment? The solute concentration inside the, so solute concentration inside the cell will be very high, okay, and the solute concentration outside the cell will be very low. So what will happen? The osmotic pressure will happen compared to the environment. Osmotic pressure. So the water movement, water will move from the lower concentration to the higher concentration. So what will happen? The cell will leads to burst, so that the cell will leads to die. So what will happen over here? The particular cell wall will give the protection for the particular cell structures or the internal structures of the particular cell. So the cell wall is a rigid structure that will give the shape and structure for the particular bacteria. At the same time, they will give the protection for the bacteria. It is like the complete outer covering that will give the protection from the osmotic pressure from the environment. Okay. So as I told you, it is made up of murin. So what are murins? Murins are peptidoglycan. Okay. So murins are peptidoglycan layers. So what are peptidoglycan layers? So peptidoglycan layer is made up of two major units, major repeats of units, that is NAM and NAG. So NAM refers to N-acetyl glucosamine, and NAG refers to N-acetyl glutamic acid. So these NAM and NAG are the repeating units of the peptidoglycans. Okay, so they will be like this: NAM, nag, nam. Okay, so there are so many repeated units like this, and they'll form the layers of the peptidoglycan, and these layers will be interconnected by the bridges of amino acids, amino acid bridges. Okay, so based on the number of layers, it will form the thickness of the cell wall. Okay, so just remember that the thickness of the cell wall means that will decide what kind of bacteria it is gonna belong. We can divide the entire bacterial world into two different classes. We all know that, gram positive and gram negative. Okay, so we are going to see the details now. Okay, so we are going to divide the entire bacterial world into gram positive and gram negative bacteria. On what basis we are going to classify these things as positive and negative? Based on a thickness and the composition of the peptidoglycan layer, and what made the particular cell wall is being thick or thinner, that is the layers of the nam and nag links. Okay, so these strings of nam and nag layer will be interconnected by the bridges called amino acid bridges, and that will form the thickness of the particular cell wall. And now we are going to see the classification, gram positive and gram negative, on what basis they are going to classify the particular bacteria. So as I told you, all the bacteria is being surrounded by a cell wall. Okay, so all the cell wall material is made up of murine, that is nam and nag repeats, and it is being interconnected by the amino acid bridges. But the point you have to remember, the major differences between the gram positive and the gram negative is the thickness of the murine or the peptidoglycan layer. Thickness of peptidoglycan layer. So how can we decide a particular bacteria belongs to gram positive or gram negative? We can go for the experiment or technique called gram staining. As you can understand, it is being discovered by a scientist called Gram, and he has discovered the staining technique to differentiate or classify the entire bacteria into gram positive or gram negative. Okay, so he has used a primary stain called crystal violet and a mordant, grams iodine, then a decolorizer, alcohol, secondary stain, anything, saffron. So these are the things we are going to use for the gram staining. So what we are going to do over here, we are going to prepare a thin smear of, so we are going to take a glass slide and we are going to prepare a thin smear. What is smear? Smear is we are going to make a film of bacteria in a particular slide. We are going to prepare a circle of film of bacteria, that is called the smear of bacteria, and we are going to make it a dry film of bacteria. Then we are adding the primary stain called the crystal violet over here, and we are going to leave it for a minute. Okay, after crystal violet we are providing a minute. So after adding the crystal violet, we are going to remove the crystal violet by washing it. Then we are adding the grams iodine. So what it will do, the grams iodine will fix the particular crystal violet to the cell's internal structures. So as we all know, each and every component of a particular biological cell will have some charge particles or charge property. So what happens over here, crystal violet have some charge and grams iodine will have a affinity property. So what will happen, the crystal violet will go and binds to the cell's internal structure evenly. Okay, so normally what is the outermost surface or outermost structure present in a bacteria? It is a cell wall. So the cell wall will take up all the crystal violet so that it will appear as a violet or the blue color. Okay, so the crystal violet will be absorbed by the particular, or it will be interacted with or it will be attracted towards the cell wall membrane, the cell wall and the cell membrane, and it will take up the color of the crystal violet, so it will appear as blue with the help of the grams iodine. Grams iodine just will fix the particular color to the cell wall. Then we are adding the decolorizer. What is the purpose of decolorizer? We are using alcohol as a decolorizer. So decolorizer, it means it is gonna remove the color. Decolorizer, it is gonna remove the color from the particular bacterial cell wall. Okay, so we are gonna add a little bit of a few drops of decolorizer along the slides of the particular smear, and that will remove the excess of colors from the particular bacterial cell. As I told you, after the addition of the decolorizer, we are going to wash it with water. Then again you are going to add the secondary stain that is completely contrast to the particular primer stain. If it is in a violet color, we are going to use a red color secondary stain. Over here it is saffronin. So the saffronin also have the same property as I told you in the crystal violet. If there is no crystal violet, the cell will take up the saffronin as a primary stain over here. So that is the principle over here. So what happens over here in case of gram-positive bacteria? We are going to classify those bacteria as gram-positive based on the property over here. So what is gram-positive? They will have a very thick cell wall so that they will take up the crystal violet very strongly, and they cannot be decolorized by means of the decolorizer, so that they will retain the primary color and they will not take up the secondary color, so that it will appear as blue. They will be appeared as blue or violet while you are observing those bacteria in case of or under the microscope. Okay, so the gram positive will appear as violet or blue. Why? Because they have very thick peptidoglycan layers or very thick cell walls, and they will take up the crystal violet and they will retain the crystal violet even though while you are treating with them with a alcohol or decolorizer. But in case of gram-negative classes of bacteria, they will have the very thin structures of peptidoglycan, so they lose up the particular stains. They lose up the primary stains of crystal violet during the decolorizing treatment. So what will happen after the decolorizer addition? It will lose the particular crystal violet color, so that it will take up the second color, that is saffronin. So the gram-negative bacteria will be appeared as pink color or in a red color. So normally while you are observing under the microscope, they will be in a pink color structures. So suppose after the gram staining, if you are seeing the blue color or violet color structures, you can say they are gram positive bacteria. After the gram staining, if you see the pink color or red color, you can say those bacteria belongs to the gram negative kind of bacteria. Okay, so what is the role of the particular cell wall? We all know how can we classify the entire bacteria into gram positive and gram negative. So we will start from the gram positive. So as I told you, what are the features of gram positive bacteria over here? They have very thick layers. And one more feature you have to remember over here, they will have some specific structures called teichoic acids. So what are teichoic acids? So teichoic acids are the components of lipids, and they will give the pathogenicity for the particular bacteria. Okay, so teichoic acid only present from the gram positive cells, and the gram negative cells, they don't have the particular structures called teichoic acid. Okay, so in case of gram negative, so they have thinner layers of the peptidoglycan, and the point you have to remember over here, they don't have teichoic acid, but they will have structures called LPS or lipopolysaccharides. So these lipopolysaccharides will make those group of bacteria, those gram negative bacteria, potential pathogens. So most of the human pathogens belongs to gram negative bacteria because of the presence of these lipopolysaccharide layers that is present in the particular cell wall of the gram negative bacteria. Okay, so these are the differences you have to remember. In case of gram positive, the thick layers of peptidoglycan and teichoic acid is present. In case of gram negative, they have thin layers of peptidoglycan and lipopolysaccharide is present over here. There is no lipopolysaccharide or LPS layer in case of gram positive. Okay, and one more thing you have to remember, in case of gram negative bacteria, they have some unique structures that is absent from the gram positive cells. They have the outer membrane. So as I told you, all the prokaryotic cells, they will have the plasma membrane or cell membrane present within the particular prokaryotic cell structures. But apart from the cell membrane, there is one more structure is called the outer membrane present in the gram negative bacteria. The point you have to remember, you are going to appear for the test. They may ask the question, the outer membrane is present only in gram negative cells, and that is absent from the gram positive bacteria. And we are going to see the next term relating with the cell wall, the periplasmic space. Periplasmic space. So as I told you, we have a cell wall. Within a cell wall, cell membrane and a outer membrane may be present. Okay, it is outer membrane, cell wall and a cell membrane or plasma membrane. So periplasmic space refers to the space in between the cell wall and outer membrane, or the cell membrane and the outer membrane. That is called periplasmic space. So almost all the virulence bacteria, they will have those virulence enzymes in the particular periplasmic space. So what is the role of cell wall? As I told you, it will give the protection and the shape for the particular bacteria. And the next thing we are going to see in the cell envelope is a cell membrane. So as we all know, cell membranes are made up of the lipids, phospholipid bilayer. But we have to remember the cell membrane is actually made up of only 40% lipids. What about the remaining 60%? The remaining 60% is made up of proteins. So actually the cell membrane is a combination of, or the mixture of, proteins and lipids. Okay. So only the 40% will make up the lipid component of the cell membrane, and the remaining 60% will be made up by the proteins. So all the proteins present in the particular cell membrane will participate in the exchanging of materials inside to the outside environment of the particular cell's environment. Okay. So that is the major function of cell membrane. So what is the function of cell membrane? It is going to participate in the exchange of materials inside and outside for the particular prokaryotic cells, and we are going to see the detailed function and structures in the next section of the particular unit, the biomolecules. Okay. Now we are going to enter into the internal structures, that is called the cytoplasm, and we are going to start with the nuclear materials. So there are DNA, RNA, plasmids. Okay. So as I told you, for all the living things and living organisms, the DNA is the major genetic materials, and they are going to inherit the characters from one generation to the next generation. And in case of prokaryotic cells, the DNA is being arranged or organized as a structure called nucleoid. So nucleoid is made up of majorly a DNA, and then it also contains proteins. Though histone proteins are absent from here, there are some other proteins involved over here that participates in the supercoiling of the particular DNA, supercoiling proteins, and we are going to see those proteins later. They are normally called gyrases or topoisomerases. Okay, so we are going to details of those enzymes and the proteins later. The point you have to remember over here, the nucleoid is made up of DNA and proteins, of course RNA. Okay, so RNA is present always along with the DNA, because the RNA is being synthesized from the DNA by means of the process called the transcription. Okay, so the DNA is the material, is in the form of nucleoid, that is the supercoiled structure. It is like a irregular structures of the particular DNA present in the cytoplasm, and RNA is present along with the DNA or the nucleoid structures. Okay, so there are types of RNAs we all know. It may be mRNA, tRNA and rRNA. So all the RNAs are present in the structures cytoplasm, and there are some extra chromosomal DNA. Extra chromosomal DNA may be present in a, you have to remember the point, it may be present based on a need. Okay, so DNA and RNA must be present for a living prokaryotic cells, and plasmids may be, or based on a need, they may be present in a particular prokaryotic cells. So what may be the role of the particular plasmids? Those plasmids gonna provide the resistance character, and they are going to secrete some substances that will give the protection for the host, that is the bacteria, from the environment. Okay, And we are going to see the details of all the biomolecules in the next section, and we are going to move to the next area over here, the mesosomes. So as I told you, the mesosomes are the invaginations of the plasma membrane or the cell membrane. So what is the purpose of mesosomes present in the prokaryotic cells? There are no mesosome-like structures in the eukaryotic cells, and specifically they are present in the prokaryotic cells, and they are participates in the, as I told you, they are increasing the surface volume. So when compared to the eukaryotic cells, the size of the prokaryotic cells is very low. I told you they are 1 to 5 micrometer in size. When compared to the eukaryotic cells, they are 100 to 10 to 100 micrometer in size. So we have to improve the surface ratio, the volume to surface ratio we have to improve, so that the structures of mesosomes will participate in the increasing the surface area of the particular prokaryotic cells. So apart from the particular role, they are also participates in the cell division and in the replication. Okay, cell division, exactly they are participating in the partition, the equal partition of the daughter cells. So during the cell division, we are going to see the cell division details later in the particular unit. So during the cell division, the cell is going to divide into two cells by means of the, so it is going to happen like this. It is a single cell and the invagination happen like this. Okay, so what will happen after this? A cell will be divided into two cells. Okay, so the mesosomes will participate in the formation of the cell division. Okay, it is an invagination of plasma membrane, and it will participate in the equal partition during the cell division, and it may also participate in the replication. During the replication, it will hold up a copy of DNA, so that it will make the equal number of DNA will be distributed to the daughter cells. Okay, so these are the roles of the mesosomes over here in case of the prokaryotic cells. So after mesosomes, we are going to see some details of the external appendages. So what are the appendages over here? So I told you, pili, fimbriae and flagellum. Okay, so only the conjugating bacteria will have the structure called pili. Remember that, only the conjugating bacteria. That means they are gonna mate by means of making a conjugation canal, so that they are gonna exchange the genetic material in between the F plus and F minus cells. So F plus, F minus refers to F plus cells, they will have the pili structure, and the plasmid called F plasmid, and F minus they will have the pilus and they don't have the structure called the F plasmid. So what happens over here while they are conjugating? So how the conjugation will happen? So there are two bacteria over here, chromosomal DNA. Okay, so this is a F plasmid. So what happens over here? Here there is a pili structure. Okay, and the pili will form a canal like this. Okay, so we are calling this canal as conjugation canal. Okay, so after the formation of canal, this F plasmid will transfer from one cell to the next cell through the conjugation canal, and it will form a new genetic material in the recipient. This called donor and this called recipient, and it will donate some part of the DNA material from this cell to the next cell by means of the conjugation canal, and we are calling this as sex mating. And the purpose of the pili is, it is going to connect the F plus, this cell is called F plus and this cell is called F minus. Okay, the F plus cells to the F minus cells, and it is participating in the conjugation. And the next structure we are going to see the fimbriae. Just remember that all the three structures are made up of proteins and similar kind of proteins. And the fimbria is shorter than the flagella. Okay, fimbria is also a kind of flagella, but they are very shorter than the flagella, and the role of fimbria is attachment to the surface. Okay, and the next structure we are going to see the flagella. So what is flagella? It is a web-like structure. It is a web-like structures present on the surface of the bacteria. Just remember that not all the bacteria will have the flagella, only the locomoting bacteria will have the structures called the flagella. Those bacteria which can move from one place to another place, they will have the structures, the appendages called the flagella. The flagella is also made up of proteins, but you have to remember that, as I told you in the last class, in case of eukaryotic cells, if they have the particular structure called the flagella, those will be arranged in a 9+2 microfilaments. But this kind of 9+2 arrangement of proteins is being absent from the prokaryotic cells. Over here it is being arranged as a just arrangement of single kind of protein called the flagellin, and there is no arrangement of the 9+2 microfilaments in case of the prokaryotic cells. Okay, so normally the prokaryotic cells, in the prokaryotic cells it is made up of three basic units: the basal unit, the ring, and the filament. Okay, so you can see that there is a basal unit and there is a ring-like structure that is gonna rotate from the internal signals, and there are web-like structure attached to the particular ring-like structure. Okay, so one more difference you have to remember over here: in case of eukaryotic cells, the movement is by means of the ATP hydrolysis. Okay, movement is by means of ATP hydrolysis. In case of prokaryotic cells, the movement is by means of the protein motive force. And we are going to see those details later in the biomolecules and in later metabolic processes. The point you have to remember over here, in case of prokaryotic cells and eukaryotic cells, there are two major differences. One thing, there are no 9+2 arrangement in case of prokaryotic cells, and there are no ATP hydrolysis happens in case of prokaryotic cells. In case of prokaryotic cells, the movement is by means of the proton motive force or the PMF, and the arrangement is made up of a single kind of protein called the flagellin. So that's all about the basic structures of the prokaryotic cells. So what we have seen today, one more thing we have left, the inclusion bodies. So what are inclusion bodies? Inclusion bodies are the storage materials. Inclusion bodies are the storage materials. So the point you have to remember over here, they are not membrane bound. So even the eukaryotic cells, they have the storage molecules, and those structures will have the membranes. But in case of prokaryotic cells, they are not membrane bound. Membrane-less structures called inclusion bodies. They are going to store the materials. For example, the carbohydrate will be stored as glycogen materials, and there are lipid-like storage called polyhydroxy butyric acids, and there are some inclusion bodies. They are going to store enzymes for the synthesis of the photosynthesis. There are so many types of inclusion bodies present in the prokaryotic cells, and we are going to see the types and the functions later. The point you have to remember over here, inclusion bodies are the storage molecules. They are not gonna cover with the membranes, and they are gonna store the particles like enzymes, carbohydrates, lipids, and in some rare cases they are gonna store some magnetic properties. Okay, we can call those as magnetosomes. So what may be the role of those magnetic particles in the inclusion bodies? They will help the particular bacteria while they are swimming in the water environment. They will help the particular bacteria to move towards the magnetic poles. We all know we are in the magnetic environment, and those magnetosomes will help the particular bacteria to move from one place to another place with the help of the magnetic property. And some inclusion bodies participates in the floating property. they will make the particular bacteria to float in a particular water environment. There are so many types of inclusion bodies with so many functions and activities, and we are going to see those things later. The point you have to remember over here from the today's class, we have seen the structure of basic prokaryotic cells, and the basic prokaryotic cell is being covered by a cell envelope. That cell envelope is made up of 3 layers. It may be cell membrane, cell wall and glycocalyx. One point you have to remember over here, in case of gram negative bacteria, one more layer is present, that is called the outer membrane layer. So outside the cell envelope there are 3 major structures, flagella, pili and fimbriae. The point you have to remember over here, not all the bacteria will have these external appendages. Only the few bacteria, only the locomoting bacteria will have the flagella, only the conjugating bacteria will have the pili, only the attaching bacteria will have the structure called the fimbriae. So apart from this, there are some invaginating structures called mesosomes. They are going to participate in the cell division and replication, and of course in the surface volume increasing the then we have seen the internal structures called cytoplasm, that is the matrix, fluid matrix present in the particular bacteria, and nucleoid or DNA structures and RNA ribosomes. We have seen plasmids and inclusion bodies. In the next class we are going to see the basic structure of eukaryotic cells, and we are going to compare it with the prokaryotic cell structures, and we are going to follow from that to the biomolecules present within each and every kind of cells, that is from the prokaryotic cells to the eukaryotic cells, and the role of each and every biomolecules in the later classes.